Good afternoon, all of you. In last lecture, I have discussed the primary cell culture, primary and established cell culture. Today, we will see or continue the same point, that is transform cell culture. The introduction of new genomic material in a cultured cell, it produces significant change in cellular phenotype, is referred as a cell transformation. I am repeating here. So, what is cell transformation? The introduction of new genomic material in a cultured cell which produces a significant change in cellular phenotype is referred or is called as a cell transformation. Then those transformed cell cultures are annulled, spontaneous or induced permanent phenotypic alterations. The consequent, the gene expression and heritable DNA changes is the characteristics of transformed cell. So it is characteristic of the cell, that is consequent gene expression and heritable DNA change. Then transformation may happen due to the spontaneous cell proliferation. The infection with transforming the viruses, then gene transcription and exposure to the chemical carcinogen and exposure to the ionizing radiations. Then the general characters of transformed cell are given. They are grouped as genetic, then structural, growth and neoplasmic. And then there are four aspects of cell transformation. First, genetic instability, then immortalization, then aberrant growth control. First, there is genetic instability, then second, immortalization, third, aberrant growth control, and last, that is malignancy. We will see one by one. First, that is genetic instability. A cell line does not always remain stable. In genetic stability, the cell line which does not always remain stable. Normal finite cell lines are genetically stable, but the continuous cells are unstable because of higher mutation rate. In vitro and mutant cells cannot be eliminated till their growth capacity is diminished. Thus, the genotypic variation also results in phenotypic variations. Second, that is immortalization. The normal cells usually have a lifespan of 20 to 100 generations, but some cells can produce continuous cell line with an infinite lifespan. Then they have growth advantage, and subsequent cultures are rapidly overgrown by the randomly growing cells. And this is called as tumorigenic cells. Then third, aberrant growth control. Cell culture from tumor and in vitro transform cultures show aberrations, that is abnormalities in growth control, and such as the growth to higher saturation densities and growth of monolayers of homologous cells. Then the cell lines slightly depend on the lower serum or the growth factor and acquires autonomous growth control by overexpression of oncogenes. Then last, that is malignancy, the cells having the capacity to generate invasive tumors. It indicates the condition of malignancy. Then a large number of properties it can be recognized in vitro transformed cells or cell culture from in vivo tumors and associated with malignant invasion. Then example of such type of transformation is tumorigenesis. Okay, the last part of our syllabus, that is application of cell culture in pharmaceutical industry as well as in pharmaceutical research. Yes, I am repeating, yes, it is the last point of our syllabus of your pharmaceutical microbiology. That is the application of cell cultures in pharmaceutical industries and also research. The cell culture is one of the major tools which is used in cellular as well as molecular microbiology and it provides excellent model system for studying the normal physiology as well as biochemistry of cell. For example, metabolic studies, then aging studies, and the effect of drugs and also toxic compounds on the cells and mutagenesis and carcinogenesis. Then it is also used in drug screening, It is also used in drug screening. Then it is also used in development. It is also used in large scale manufacturing of biological compound, for example vaccines and therapeutic proteins. Then the major advantage of using the cell culture for any of this application is the consistency and the reproducibility of result that can be obtained from using a batch of clonal cells. Then there are different applications. First, in model system, then vaccine production, third toxicity testing, then next cancer research, next virus cultivation and study, the next cell based manufacturing, then gene therapy, next genetic counseling, then genetic engineering, next drug screening and development, then immunological studies, and last of all I am repeating the applications of this. First application, that is model systems, then vaccine production, third toxicity testing, fourth cancer research, fifth virus cultivation and study, sixth cell based manufacturing, seven gene therapy, 8 genetic counseling, 9 genetic engineering, 10 drug screening and development, 11 immunological studies, and last that is others. We will see one by one. Model system. In practical, the cell cultures act as a prototype for study about the other cellular activities. Those includes first understanding of cellular biology as well as biochemistry, then next cellular interaction with virulent agents, then cellular reaction toward the cells, then process of cell aging and its triggers, then cellular nutrition, then second vaccines production. The one of the most important use of cell culture is in research and production of vaccine. Then the ability to grow a large amount of virus in cell culture eventually led to the creation of the polio vaccine. Then the cells are still used today on large scale to produce vaccine from many other diseases like rabies, chickenpox, and hepatitis B. Then, in early times, the researcher had to use a live animal to grow the polio virus, but due to the development of cell culture technique, they were able to achieve much greater control over virus production and on the much larger scale, which eventually developed vaccine and various treatments. Then, however, the continuous cell lines are not used in virus production for human vaccines as these are derived from the malignant tissues or possess malignant characteristics. Then, third, toxicity testing. The animal cells are being used for testing various drugs for its toxicity level. Then, cultured cells are widely used alone. The cultured cells are widely used alone or in conjunction with animal test to study the effect of new drugs, then study the cosmetic and the chemicals on the survival and the growth in wide variety of cell types. Especially important are liver and kidney derived cell culture. Then next use or application, that is cancer research. The cultured cancer cells are being used as a biological system for determination of the suitable drugs or dose which is selectively destroy a particular type of cancer cells. Then thus the cell culture is an effective tool of cancer research. See here I am repeating, the cell culture is an effective tool of cancer research. Though the normal and cancer cells are grown side by side, it is easy to differentiate and study those cells easily. Then, even nowadays, the mechanism of conversion of normal cells to cancer cells can be studied with cell culture. And this conversion may include a chemical, viruses and radiation. The next application, virus cultivation and study. The cell culture is widely used for the propagation of viruses as it is convenient, it is easy, it is economic, and it is easy to handle compared to other animals. Then it is easy to observe cytopathic effects and also easy to select particular cells on which viruses are grown as well as to study the infectious cycle. Then the cell lines are convenient for virus research because cell material is continuously available. The next, the continuous cell lines have been extremely useful in cultivating many viruses previously difficult or the impossible to grow. Then cell based manufacturing. The cell-based manufacturing is comparatively new and cost-effective approach. This approach is employed in production of wide variety of pharmaceutical important product like large-scale production of viruses for use in vaccine production, or for example polio vaccine, rabies vaccine, chickenpox vaccine, and hepatitis B vaccine. Then genetically modified or the engineered production in large scale is another approach by the animal cell cultures. Then these products include various pharmaceutically and medicinally important proteins, monoclonal antibodies, and insulin, hormones, which all have huge commercial values. Then another important area of cell-based manufacturing is in replacement tissue and organ therapy. Then by using the cell culture, artificial skin for use in treatment of burn and ulcers is the first commercially available cell-based product. Then on this way, the next, that is gene therapy. Cells having functional gene and it can be replaced to cells which are having non-functional gene, and for which the cell culture technique is used. The next, genetic counseling. The genetic counseling is an important indirect application of cell culture. Fetal cell culture extracted from pregnant women, it can be used to study or examine the abnormalities of chromosomes, genes using karyotyping. In this finding, it can be used in early detection of fetal disorders. Then, genetic engineering, animal cell culture are used to produce commercially important genetically engineered proteins such as monoclonal antibodies, insulin, hormones, and etc. Then cell culture enables the cellular processes of gene expression of reprogrammed or genetically manipulated new genes. Then this technique can also be used to produce new proteins in large quantity in cultured cells. Then cultured animal cell can be used to introduce new genetic material like DNA or the RNA into the cell. Then production of genetically engineered protein from insects by genetically manipulated baculoviruses is a significant contribution of the cell culture. Next application, drug screening and development. The cell-based assays are currently gaining importance for the pharmaceutical industry for the drug screening as well as drug development. Then for the area of new drug development, the animal cell cultures are used to study the cytotoxicity of new drug. Yes, where it is used to study the cytotoxicity of new drug. This is also used to find out effective and safe dosage of new drugs. Then even new drug development, high the put screening of compound plays an important role that may have the key potential drug use. The next application, immunological studies. The cell culture technique are used to know the working of various immune cells, cytokines, working of various immune cells, cytokines, lymphoid cells, and interaction between the disease causing agent and host cells. Then last application in order, the cell lines are also used in vitro fertilization, that is IVF, in vitro IVF technologies, recombinant protein and p-drug selection, and also improvement. So this is all about the applications of tissue culture, or the applications of cell culture, applications of cell cultures in pharmaceutical industry as well as research industry. Here your syllabus is your syllabus of pharmaceutical microbiology is over.